Recommended Ipsilateral initial and booster vaccination. A clinical trial participant receives a dose of an investigational Ebola vaccine at the National Institutes of Health Clinical Center. Primary vaccination and subsequent boosters for a given infectious disease injected into the deltoid muscle of the same arm in adults is an ipsilateral vaccination productive of a stronger protective immune response than would result from contralateral primary and subsequent booster vaccination involving both arms in adults. Ipsilateral secondary boosting is relatively more productive, inducing a stronger immune response, since it provides for drainage by the same lymph nodes involved in the physiology of immunological priming. The ipsilateral lymph nodes on the side of such vaccination were observed to be notably larger in size and capable of higher metabolic function compared with those observed after contralateral arm vaccination. It is believed that this sequential vaccination protocol for some infectious diseases may be capable of producing a stronger immune response, conferring more protective immunity in certain respects to future disease exposure when delivered ipsilaterally in the same arm than would be the case upon contralateral administration. Cluster of differentiation, CD cell surface proteins of assigned number referred to a particular cell phenotype and function. Spike glycoproteins project from the outer membrane of the virus cell that bind to the host cell receptor, facilitating viral entry and viral infection of the host. Ipsilateral vaccination has been found to result in relatively higher protective virus-neutralizing spike protein-specific antibody response rates, spike protein-specific CD8 T cell responses, as well as spike-specific CD4 and CD8 T cells respectively functioning in humoral immunity and viral cytotoxicity. B cells produce infection-fighting protein antibodies that function in the adaptive immune response responsible for humoral immunity. Ipsilateral vaccination has been observed to result in relatively higher amounts of B cells and high avidity B cell receptors, enhancing interaction with the antigen and supporting T helper cell function. Two T cell types function as cytotoxic destroyers of infective cells, as T cytotoxic cells and as T helper cells that signal other immune cells to fight infection, functioning in the adaptive immune response. Spike-specific immunoglobulin G antibodies are more strongly induced upon secondary booster vaccination, with virus neutralizing activity relatively higher than is the case involving contralateral booster vaccination observed in a COVID-19 vaccine study in a SARS-CoV-2 naive population. Cytotoxic T lymphocyte-associated protein, CTLA4, also referred to as CD152, is a protein receptor functioning as an immune checkpoint for the downregulation of immune responses. Subsequent to T cell activation, CTLA4 is present in the intracellular vesicles, becoming available to the immunological synapse formed between T cells and antigen-presenting cells, inhibiting further activation of T cells by blocking signals initiated by T cell receptors and CD28. CD28 is a T cell protein productive of the co-stimulatory signals of T helper cell activation and survival. In combination with T cell receptors, it signals for the production of certain interleukins that are glycoproteins of immune response regulation. Although spike-specific CD4 T cell levels were not dissimilar upon ipsilateral or contralateral vaccination, markedly higher CTLA4 expression was observed subsequent to contralateral vaccination. Mark Siegel, MD, is a New York University Langone Medical Center internal medicine specialist, professor of medicine, published medical author, and media commentator who states in this regard, quote, "It makes sense, however, that the pump is primed better by going back to the limb that the body is used to, and that the immune response and inflammation from the shot could be more substantial." Please pause and enlarge the next 3 images for detailed study. 4 broad groups of vaccines. Messenger RNA vaccine mechanism of action. DNA vaccination. DNA-encoded antigens are transcribed, translated, and presented on major histocompatibility complex 1 and 2 molecules in vivo, promoting robust anti-target immunity. New presentations coming soon. Thanks for watching. Thank you to current and new subscribers.